Hello, everyone, and welcome to today's webinar. My name is Olga Bodie, and I'm a SYNGAP parent and volunteer here at SYNGAP Research Fund. And our presentation today is linking SYNGAP1 with human-specific mechanisms of neuronal development. And I have the pleasure to introduce today's speaker, Dr. Pierre van der Hagen. Dr. van der Hagen is a full professor at the Department of Neurosciences in the Faculty of Medicine at the University of Leuven in Belgium. He also serves as a group leader at the VIB KU Leuven Center of Brain and Disease Research. Dr. van der Hagen received his MD in June of 1992 from the ULB Medical School and his PhD in biomedical sciences in 1996, also at the University of ULB. He completed his postdoctoral fellow at the Department of Cell Biology at Harvard Medical School, where he attended from 96 to 2000. A recorded version of the webinar will be available on the SRF website under webinars on the family menu. And by the end of this presentation, you will have the opportunity to get the answer to your questions. We'd love to hear from you. Please write your questions in the Q&A below. And for those of you just joining us, welcome. And again, our speaker today is Dr. Pierre van der Hagen. His presentation today is linking SYNGAP1 with human-specific mechanisms of neuronal development. It's now my pleasure to turn things over to Dr. van der Hagen. Thank you.
Awesome. Thank you so much, Olga. I hope everybody can hear me. Mm-hmm. And it'd be useful for me to make sure that everybody can also see my presentation. Can you confirm that maybe, Olga, that you see?
Yes. Mm-hmm.
Yep. You do? Okay. Do you see my mouse as well?
Yes.
Yes.
All right. Fantastic. Okay. So, well, thanks a lot for the invitation. It's a real pleasure for me to be able to speak to you. And I guess it's quite remarkable, I guess, that new technologies enabled us to do this. Only a few years ago, it would have been probably basically impossible. But despite, yes, I think the fact that we all prefer live presentation, I think this is a wonderful opportunity. I hope to share with you some of the work we're doing, and I would be very interested also to hear your feedback and your questions. So I realize that I'm talking here to an audience who's very interested in disease associated with SYNGAP1 deficiency. This is not the focus, the main focus anyway, of our lab. And in fact, the focus on our lab is neither neurodevelopmental diseases. It is actually more a very basic question that has to do with the origin of what makes our brain human. So a question very much related basically to the development and to the evolution of the brain in our species. But what I'll try to show you today is that there are in fact quite intriguing links between, on the one hand, the mechanisms that somehow link development and evolution of the human brain, and on the other hand, that link these with neurodevelopmental diseases such as the one caused by mutation in the SYNGAP1 gene. And I think beyond, let's say, the curiosity-driven aspect that I will show you as a scientist, I think you will find out, hopefully, that there are interesting, potentially interesting implications for a better understanding of neurodevelopmental diseases, including SYNGAP1 pathy, so to speak. So, without further ado, let's start with a short introduction. What I like to remind or explain to some of you what we know basically and what we want to understand about what makes our brain human. So there are multiple levels of specificities of the human brain when you compare it with the brain of other species, including the chimpanzee, so our closest cousin on this planet. And among other things, there is, of course, the fact that the human brain is much expanded in size, and this is mostly linked to the increase in size of the cerebral cortex that contains many more neurons, but also not any type of neuron, but mostly those neurons here that populate the surface of the cortex, the upper layers, and that are specialized actually in what we call cortico-cortical connectivity, that is basically intracortical circuitry that is thought to underlie a lot of the higher cognitive function that are characteristic of our species. There are a number of things that we could spend hours discussing what makes the brain human in terms of anatomical, molecular, and physiological differences. But the one I really want to focus today upon is the fact that it is characterized, the human brain, by a very prolonged development, which is also called human brain neoteny. And what I mean by that is illustrated to you here on the left side. It's the fact that basically it takes up to 2 decades for the normal human brain to develop. And you can see here morphologically some very striking images where you can see on the one hand how the brain of a 1 month old is increased in size over, in this case, up to 6 years. And this is not linked to an increase in the number of neurons, because the neurons were already generated before birth. This is mostly linked basically to the growth in size and complexity of the neurons themselves, in particular growing dendrites, axons, synapses, and so on. And you can see here, for instance, that in the prefrontal cortex, up to 15 years is probably needed to get to the maximum amount of synapses that can be generated. You can see here the comparison with the mouse, where basically the same process that we know in the human takes up to 2 decades, takes about 6 weeks. So that's what neoteny is about. It's the fact that there is a sort of out-of-scale prolongation of the development of the human brain, and most strikingly of the cerebral cortex. And you may wonder, is that good for anything? Well, it's actually good for many things potentially. And in fact, it's been recognized for decades, if not more. I think even Darwin mentioned this, that it's probably a selected trait, as we say in evolution. So something that was somehow selected in the evolution of hominids and then pre-hominins and the human species, because basically the prolonged development of neurons after birth and for many years allows basically for a longer period of what I would call, to simplify, an optimization of the neural circuits in response to the environment. So this prolonged development basically makes our brain uniquely sensitive for a very long period to the environment, social interactions with the outside world, and so on. And this is indeed, as we all know, a critical feature of human brain development. Now, on the other hand, even though we would recognize that at least for the evolution, this is a very important process, there's very little known about it. So it's still very little that is understood about how is it that human brain development is so much prolonged. And on the other hand, and so that's one first thing that we're very interested in our lab, is to try to understand what are the mechanisms, molecular, cellular, genetic, that somehow enable to understand how come the human cortex is so neotenic, makes so much time to develop. And on the other hand, what we're also very interested in is what are the implications of this neoteny for normal brain function, and then of course also for brain diseases. Because if something is really important for normal brain function, it is reasonable to hypothesize that when it is disrupted, it may be linked, it may cause actually impaired brain function, diseased brain function. And so these are the two aspects that we study a lot in the lab, and where, as you guess, this is basically what brought us to study, to get interested into SYNGAP1. But before we get there, I want to first tell you a little bit about how we got interested into the mechanisms underlying neoteny and what we know about it at this point. So the way we got interested in this is first, a number of years ago when we and others invented ways basically to make cortical neurons, so make neurons of the cerebral cortex artificially in a dish from pluripotent stem cells. And I'm sure many of you are familiar with that type of technology. But what we did basically to try to probe basically the function of these human neurons is to transplant them in the mouse, to basically test to which extent, found in the context of an in vivo brain, these neurons generated artificially could develop. So it was really, to start with, just a sort of technological tool. And that's just to show you a few images of these human neurons that we inject in the ventricule of the brain, and then we can see them migrate here. These are the transplantation are done in neonatal mice, so young, very young pups. And then if we wait a few weeks, you can see now that the mouse cortex is stuffed literally with a lot of human neurons that you see here in green, and that these are the neurons that we call xenotransplanted because they come from another species, in this case, the human species, and they go into the mouse. Now, what's good about this, despite the fact that it's an artificial system, in fact, it revealed to us something quite relevant to what I was telling you about the neoteny. And that's what I'm showing you now. What it revealed to us is basically the following. If we take mouse pluripotent stem cells and differentiate them in cortical neurons in addition, transplant them in the mouse, well, it takes them about 4 weeks, as you can see here, to become in the mouse relatively mature cortical neurons. If we do the same experiment with human neurons transplanted in the mouse, that's what's shown to you here, you see that it takes many months. In fact, it takes more than a year to get neurons that start to be mature. And so that's strikingly reminiscent basically of the very prolonged timing of neuronal development. And we and others since then have done many experiments taking, for instance, neurons derived from stem cells of different species and transplanting it in the mouse. And the bottom line is what I show you here, which is that the developmental timeline of cortical neuron following transplantation from any species into the mouse is remarkably similar to the time it takes for the neurons of that species in the brain of these species. So in, for instance, the chimpanzee, the human, the macaque, or the mouse. So strongly suggesting that basically there's probably a sort of clock or hourglass, as I prefer to say, that is intrinsic to the neurons and that controls the developmental tempo of the cortical neurons. Now the question is, what could be this developmental tempo? And what is this hourglass or this clock that's in the neurons? And so I will get that into a moment, but before that, I want to show you one last important aspect that make up your mind, and which is that maybe these human neurons here develop very slowly in the mouse, not so much because they follow an intrinsic developmental clock, but because they're just not in the right place, and so that they are wrongly developing, and because of that, they just take they just slow down and maybe stalled in some kind of aberrant developmental path. But in fact, we spent many years to test that, and the answer is no. These human neurons actually seem to develop quite normally. And I think the best proof we have so far along these lines is shown to you here. What we've done basically in the lab is to transplant the cells, and then wait many months for the neurons to develop, and then probe the function of these neurons using in vivo imaging. And in this case, the experiment that I'm showing you is basically an experiment of microscopy multiphoton imaging of the mouse cortex of a live mouse that is awake. And you can see here that we can image individual human neurons in that mouse. And we are here located in the visual cortex, and the mouse at that moment is watching visual stimulation, and we can look at the response of the human neurons. That's what's shown to you here. You can see on the upper left what the mouse are watching, what the mouse is watching, and you can see the individual neurons. And I hope you can see two things. First, that the neurons respond to the visual stimulation, and second, that they don't respond all at the same time. They are all specifically responsive to specific stimuli corresponding to different directions or orientation of the visual stimulus. And that is a characteristic feature of the visual cortex. So to us, this strongly indicates that these human neurons, despite the fact that they develop very slowly, are actually, in the end, acquiring very mature and sophisticated circuit features. And you will see that this is providing us novel tools to study human neurons in vivo and understand what could be going on during normal development as well as in disease. So to summarize the first part, there is an intrinsic hourglass of development of human neurons. If we transplant the human neurons in the mouse, they develop at their own pace for that reason, yet they can achieve substantial maturation and functionality. And so one question that we're interested in the lab, as I told you before, is what are the mechanisms that regulate this timing? And then on the other hand, what could be the implication for normal or abnormal development in the human brain? There's a third implication that I will not have time to discuss with you, but that I'm happy to discuss later on in the Q&A, and which is the perspectives that this type of work is offering for potential therapy leading to brain plasticity or repair. Now, the mechanisms, I'm going to go quickly here because this is really hardcore developmental biology, but basically you could imagine two main mechanisms. On the one hand, you could imagine that the genes that control the development of the neurons in the human and the mouse are following different timelines or are actually different genes, and this is why it's taking a different time in the end. Alternatively, there could be global changes in the neurons that somehow influence the same genes along a different speed. So that's what I mean by changes within the gene regulatory network or outside of it. And what we found in the lab is that the two hypotheses are true. The two types of mechanism coexist and in fact work together to control the hourglass of the developmental timing of the neurons. What we found a few years ago, yes, but that we published a few weeks ago, is that, for instance, the basic metabolism of the neuron is very important as a global controller. If you're interested, you can check our paper that came out recently in Science on this. But there are also gene regulatory mechanisms and new genes that are at stake. And that's what I'll be telling you about now, in particular of a new gene family, meaning a human-specific gene family that we think is a very important controller. Now, what do I mean by new genes? So many decades ago, developmental biologists in particular, as well as geneticists, found out the very surprising thing that I'm sure you all know, and which is that the genomes of many different species contain the same amount of genes and are very similar to one another. This is true vastly, meaning that, for instance, between human and chimpanzee, most of the genes are common, but there are a few genes that are different. And these basically come from what we call gene duplication, meaning one gene can become another one through a copy-paste mechanism and then change through evolution of function and structure. And these genes that we call human or hominid specific, there are many of them. And a few years ago, we identified several dozens of them that are expressed in human cortex development. And in particular, among them, some genes that you can see here that are selectively upregulated during human cortex development in cortical. Now, what are these genes? These genes were actually discovered by our colleague and a friend, actually, Frank Polleux, who works at Columbia University. What he discovered a couple of years ago now is that there is a gene that is common to all mammals, so also present in the mouse, that's called SRF2A. And then in recent evolution, a few million years ago, in our ancestors, this gene was duplicated into other genes called SRF2B and C, which we found to be expressed heavily in cortical development. And what Frank and his team also found, and that you will see is very relevant to what we're discussing today, is that if you overexpress the human-specific gene in the mouse, it results in a delayed maturation of the mouse cortical neurons. So a kind of neoteny. Of course, not transforming the mouse neurons into human cortical neurons, but nevertheless, decreasing very substantially the speed of their development. And interestingly, it's also important for what I'll be telling you about. They saw the same thing if they were removing one copy of the SRF2A gene, the ancestor gene, so suggesting that these human-specific genes act as inhibitor of the gene from which they derived, and that is present in all mammals. Now, this was a number of years ago. What we set up to do then is to study whether these SRF2C gene present only in the human genome to understand what they could be doing in human neurons. And that's the work of a very talented postdoc in the lab, Baptiste Libet-Philippot. So how did we do this? Well, we first used our system, starting from stem cells, transforming them in cortical neurons, and then infecting them with these constructs that lead to downregulation, in this case, selectively, of the SRF2C gene. You can see here the SRF2C and how it goes down following this knockdown. And then the setup is basically to first knock down the gene, then transplant the human neurons that no longer have the human-specific gene SRGAP2C, and then ask how they develop in the mouse in vivo. And looking in particular, in this case, at the morphological development, look at these dendritic spines that reflect synapse formation, and then also look at their physiological activity, again, reflecting in this case synapse formation as well as maturation. And what we found is a very striking observation, I think. So you can see here basically how these dendritic spines develop over time from very few spines to many in control situations. You see it takes here up to 18 months to get the plateau of development of the dendritic spines. So reflecting, of course, the neoteny I was telling you about. And now here is the situation following the loss of the human-specific gene. You see that the dramatic increase in the number of dendritic spines over time. And that's what's shown to you here. So here is the normal pace of development of the dendritic spine increase. And you can see here it's accelerated following the loss of the human-specific gene. Conversely, if we remove the ancestral gene, the gene that's present in all mammals, SRGAP2A, it's the opposite phenotype. We see fewer dendritic spines, which fits with what Franck Peleux initially suggested, that these genes, SRGAP2A and SRGAP2C, are antagonists with one another, and that the human-specific genes that we see here are actually required for the normal neoteny of the human cortical neurons in vivo. We confirm this using physiology. And again, same thing, I'm going to go quickly here, but you can see increase in the frequency of the synapses, in the amplitude of the synaptic currents, and the frequency of the synaptic potential, sorry, and their amplitude. And you can see here also changes in the ratio of AMPA and MDA currents that reflects basically increased maturations of these synapses. So overall, there's an acceleration in the formation and in the maturation of the synapses in the cortical neurons that lack the human-specific genes. So that was already quite interesting for us because it was the very first demonstration that a human-specific gene, so a gene present only in our species and no other species, is actually required for something that we think is quite important because it's partly basically setting the very slow tempo of human cortical neurons. Of course, this is just one gene among probably many others that contribute to it, but it was a first important step to try to move on. And so to try to understand which other genes could be involved in this, we asked basically this classical question in developmental biology, which is what are the downstream mechanisms, which are the other molecular players that together with the SRGAP2C could control the neoteny. And you will see so how we did this. A first approach that we use is basically, again, doing a knockdown of the SRGAP2C human-specific gene of the human cortical neurons generated from the stem cells, but then staying in the Petri dish for practical reasons, then look at the composition of the synapses of these neurons. And what we first found, as you can see, is that overall, the compositions of these synapses is fairly similar at the molecular level with this very gross approach, whether we knock down SRGAP2C or SRGAP2A. And by the way, I'm sorry, I should have started with this. You can see here the level of SRGAP2A, so the ancestral protein. And you can see that the level of this protein is, of course, going down when we use a knockdown to knock down SRGAP2A. But you can see, interestingly, that it goes up when we knock down SRGAP2C. And this was actually sort of expected. There is data from other labs that have gone beyond the observation that SRGAP2C and SRGAP2A seem to that SRGAP2C seems to inhibit SRGAP2A. It seems that this inhibition goes through a diminution of the amount of SRGAP2A, meaning that if you remove SRGAP2C, well, then you increase the level of SRGAP2A. Okay, now how about other types of proteins? And this is where, I guess you guessed it, we stumble upon SYNGAP1 that turned out, sorry, I'm going a bit quick here, that turned out to be actually dramatically changed following knockdown of SRGAP2C. So you can see the normal levels of SYNGAP1 in the cortical neurons, and you can see how it's decreasing following removal of the SRGAP2C human-specific gene. And you can also see already here, we'll get back to that later, that the levels of SYNGAP1 seem to increase, on the other hand, when we remove SRGAP2A. So since C is blocking A, we had this model here suggesting that C is blocking A, and A is maybe diminishing the levels of SYNGAP1. And this model would explain basically what you see here. So that to us was very interesting. And so we asked the question, of course, of what could be the relevance of the biology of SYNGAP1 in human neurons. And now we get in a territory that I think many of you are much more familiar, and maybe to some extent more familiar than me, basically of the wealth of knowledge about SYNGAP1, that on the one hand, from a basic perspective, is a very abundant postsynaptic protein involved in synapse development and function, the mutation of which can lead to intellectual deficiency, epilepsy, autism spectrum disorders. And interestingly, the work of Gavin Rumbaugh and colleagues has shown a number of years ago, mimicking the mutations found in the patients, but in the mouse, that it can be associated with accelerated development of the synapse in the mouse. So a little bit conceptually similar to what I showed you with removal of the human-specific genes in the human cortical neuron. So we thought that this was maybe more than a coincidence. And so we looked now, in parallel, I would say, to the study that we did on the human-specific gene, we looked now at the SYNGAP1 gene, but in the same model in human cortical neurons. And this is a work that we did in the lab, thanks to Ben Vermark, a very talented postdoc, as well as Vincent Bonin, a visual physiologist, because you will see that we went actually, when it comes to SYNGAP1, all the way to visual function and how the human neurons can be implicated in that in normal and SYNGAP1 mutated conditions. So first, we generated SYNGAP1 mutant pluripotent stem cells and cortical neurons using conventional CRISPR-Cas technology. You can see here normal neurons and the level of SYNGAP1 here, what we would call knockout neurons. So with no protein at all. And here, SYNGAP1 haploinsufficient neurons, as I will call them. So when one copy is deficient while the other one is normal, which mimics basically the disease. And what we've done then is to prepare cortical neurons of each of these three genotypes and then differentiate them in cortical neurons, transplant them in the mouse, and compare them in terms of morphological and synaptic development, as well as looking at them at the level of the circuit in the visual cortex. So that's just to show you basically the type of imaging that we can do in the mouse of the human neurons, but in this case, looking at the dendritic spines. And one important thing is that since we can image those neurons in live animals, we can repeatedly image the same neurons over time and really have an idea of the dynamic of development of these neurons, which is really critical for the question we're interested in of developmental timing. So you can see here similar neurons over time, and you can see in normal conditions how the spines increase in density, and you can see in the SYNGAP1 heterozygotes and homozygote mutants how the dendritic spine basically formation is accelerated. You can see here a first detailed quantification, and here a synthesis of that, where you can see that the knockout SYNGAP1 neurons basically are already very high, very early on, a few months after transplantation. And you can see that the haploinsufficient neurons are also actually increased in speed, quite importantly compared with the control. So there's a striking acceleration of dendritic spine formation in the human cortical neurons in vivo. And this is actually quite consistent with the data that Gavin Rumbaugh and colleagues had found in the mouse. With one important thing to bear in mind is that in this case, what we found is basically that the mutant human neurons are basically months ahead in development compared with the control, while in the mouse, because of their very fast speed of development, the difference of time was only a few days. And I think it's something important to bear in mind. Now, this is for the morphology. This is now for the synaptic development. And you can see here again that there is an increase in the number of the synapses as well as in their maturation following the loss of one copy of SYNGAP1, so in the haploinsufficient condition. So consistent with the fact that synapses are formed at a faster speed and also they mature faster in the SYNGAP1 haploinsufficient neurons. Finally, the third thing we looked at in the SYNGAP1 mutant neurons is potential consequences at the circuit level. And I think that's a very important level, of course, especially when one wants to understand brain function and disease, because we are not just made of, our brain is not just made of a couple of neurons, it's made actually of networks of these neurons. And these are the ones that subserve specific function. So if we want to understand the links between a particular genetic defect and disease phenotypes and go to diagnosis and treatment, we really need to basically study this circuit level. And so that's what we did using the approach I told you before of using mice that were transplanted and where the human neurons are infected and transduced with genes that enable in vivo functional imaging in live mice that are then stimulated with specific visual stimuli and ask how the human neurons respond, human neurons with a normal genotype or that are displaying SYNGAP1 haploinsufficiency. And we made two observations that I'm sharing with you now. The first important observation is here when the mice are not looking at visual stimulus. So they're basically stimulated with a gray screen so that there's not much stimulation of the visual cortex. And you can see there's a first important difference between the SYNGAP1 haploinsufficient and control neurons, which is that their level of activity is increased. And I think that may have very important pathological implications, obviously. You can see here the quantifications. So this suggests already, probably because of the increased speed of development and maturation of the synapses, that these neurons are more active than they should at that stage of development after 7 months post-transplantation. On the other hand, and perhaps even more strikingly, we looked then at how these human neurons are able to respond to visual stimuli. And I'm not going to go into the details, but you can see and guess here that we can look at various neurons and see how they respond to particular stimuli that again correspond to specific directions or orientation of visual stimuli. That's the way visual physiology uses and classifies the neurons. And we can see basically that the SYNGAP1 haploinsufficient neurons respond actually much faster in time, meaning that while at 2 months post-transplantation, there are very few cells in normal conditions that can respond. That's because they're neotenic, they're very slow to develop. We can already see that SYNGAP1 haploinsufficient neurons, a substantial fraction of them can respond, and this goes on with time. So suggesting that what I would call the acquisition of visual responses is also accelerated in the human haploinsufficient neurons. And we are exploring this further now to look at further consequences of this, but I think this is all consistent with what I've been telling you in the beginning, that the human neurons follow intrinsically their own pace of development, depending on their species. And so in that case, the human neurons develop very slowly, but it also depends then on the genome composition of these neurons. And if SYNGAP1 is, if one copy of SYNGAP1 is missing, well, these neurons now start to develop faster, again, in an intrinsic fashion, independently of the environment. You can also see another important thing here, which is that here what we call, for instance, orientation selectivity index, which is basically an idea of the precision of these neurons to recognize specific stimuli, is actually similar between the neurons of the two genotypes. So suggesting that it's really the speed of development of the neurons that is specifically altered in these conditions in the human neurons at that early stage of development. Now let's go back to the molecular mechanism, and this will be the last part of the talk, where I will try to basically link again the observation I showed you before, that SRGAP2C is this human-specific gene involved in neoteny of human neurons, and then SYNGAP1 for which I showed you that it's also involved in the neotenic speed of development of human neurons, and SRGAP2A that could be potentially in between from the biochemical data I showed you here. So we did another experiment, and in fact, I'm showing it to you now. And that experiment is to look now at the molecular composition of the synapses in the neurons that are haploinsufficient for SYNGAP1 first. And what you can see here is that the levels of SYNGAP1 are lower. That's expected. There's one copy missing. But you can see something else here, which is that the levels of SRGAP2A are increased when we remove one copy of SYNGAP1. So that means that the model I showed you here is actually only partially true, or let's say it's only part of the truth. In fact, if you put everything together, it means that SRGAP2A somehow is downregulating the level of SYNGAP1, but what is shown to you here is that SYNGAP1 is downregulating the level of SRGAP2A. So it's as if there was a sort of tug of war, if you will, between the two proteins to be located at the synapse, and somehow each one is inhibiting the other one, with SRGAP2BC being upstream of that and tipping potentially the balance towards one of the two sides. And the second interesting experiment that we did, at least I think the result is interesting, is that in the SYNGAP1 haploinsufficient neurons, we then removed SRGAP2A, and reasoning that this should have an impact on SYNGAP1. And indeed it has, and you can see the impact is quite dramatic. So in the SYNGAP1 mutant neurons, where SYNGAP1 is much reduced in quantity in the synapses, when we also remove SRGAP2A, you can see that now it goes to levels that are almost normal. And so that's what triggered then a question that I showed to you, the next question that I'm showing you here, and which is basically whether SRGAP2 downregulation could somehow rescue the SYNGAP1 synaptic phenotypes I was showing you. And so to address that, oops, I'm going a bit fast, yeah. What we did is to take cortical neurons that are deficient in SYNGAP1 or normal for SYNGAP1 levels and knock down the levels of SRGAP2A and ask what's the outcome now in terms of morphological and physiological development. And here is the first type of experiment. So this is to remind you again what I showed you before, which is that the density of the dendritic spines is increased when we remove SYNGAP1. But now here is what's going on when we remove SRGAP2A at the same time. You can see that basically those levels go down to almost normal levels. That's what's shown to you here also. You can see the levels going up following SYNGAP1 mutation and how they go down following the double removal, if you will, of SYNGAP1 on the one hand and SRGAP2A on the other. I think it's even more striking if you look at the physiology, where you can see again normal SYNGAP1 neurons and heterozygotes neurons, and you can see that basically the SYNGAP1 normal neurons show an increase in the synaptic frequency and amplitude following the haploinsufficiency of SYNGAP1. And this is basically going back to what seems like normal values, or at least much reduced and closer to normal values following the knockdown of SRGAP2A. And again, the same is true for the AMPA and MDA ratio. So suggesting that the increase in the speed of the dendritic spine formation and maturation and of the synapse formation and maturation is basically decelerated when both of the genes are reduced. To wrap up and try to conclude on this, on the one hand, we have this molecular model here that I show you where there is those three proteins interacting with one another in a very dynamic way in the human cortical neurons. And that's basically to show you the way we think it's going on. So on the one hand, in non-human neurons, in the mouse neurons, for instance, SYNGAP1 and SRGAP2 are involved in a tug of war where they inhibit one another, and that somehow leads to a relatively fast speed of maturation of the neurons. In the normal human neurons, the human-specific gene SRGAP2C is actually tipping the balance by inhibiting SRGAP2A to reduce those levels and thereby basically increasing the neoteny, slowing down the speed of the human cortical neurons. While in the case of SYNGAP1 haploinsufficiency, where the levels of SYNGAP1 are now lower, well, this tip of balance basically is going the other way around, so that the development of the cortical neurons, the human cortical neurons, is now accelerated in an abnormal fashion. So if we want to wrap up in a more conceptual way, what I showed you today is on the one hand that the tempo of synaptic development is controlled by a balance of SRGAP2 and SYNGAP1 genes, that this balance is somehow tipped by a gene that's only present in our genome, and then that, to our surprise, and I think it's interesting, obviously, for the understanding of the mechanisms underlying disease caused by SYNGAP1 haploinsufficiency, SYNGAP1 seems to be directly linked to this mechanism that controls the speed of development of the human cortical neurons and of this human-specific neoteny. So with that, I will stop and thank again the people involved in this work. I mentioned Baptiste and Ben, as well as Vincent Bonin. I would like to mention also Rio Iwata, who was a driving force of many of the experiments that were shown to you today. And I forgot to mention that many of the experiments that we did on the SRGAP2C human-specific gene were actually done in collaboration with our long-standing collaborator, Franck Polleux, and his former postdoc, Cécile Charrier, now in Paris, who discovered originally this gene in the human genome. So with that, I will stop, and I will be happy to take any questions or hear your comments. Thanks a lot.
Thank you. Thank you very much. That was absolutely fascinating. These webinars, generally, we have a few parents and a few scientists join, but then parents watch them later. So what I try to do is pepper you with as many questions as I can anticipate. And I know we have other parents and people on. So if you have questions, please put them in the Q&A or raise your hand if you want to talk. But I mean, the obvious question is, great, in human hets, can we knock down SRGAP2A and recover the phenotype, which I think gets to your third point about juveniles and plasticity that you didn't have time to touch on. So if that's an accurate sort of simple hypothesis, what are the risks of knocking down SRGAP2A? I mean, is that actually feasible in a living creature? What else would go wrong if you did that in a SYNGAP het human? And then could you tease a little bit what you didn't talk to today, which is the rescue and the plasticity? Because those of us who are parents of human hets are living, waiting with bated breath for ASOs or gene therapies to upregulate SYNGAP. And then the question is, okay, what is that? Then we're going to create an entire new class of disease, right? 0 to age X, haploinsufficient, recovered via gene therapy. What do those brains look like where you reintroduce SYNGAP later in life? So just 2 softballs for you, 2 easy questions.
These are, 2 fascinating, 2 very important questions on 2 fascinating topics. Obviously, I don't have straightforward answers for you. On the one hand, I think what would be expected in development if you knock down SRGAP2A is the opposite phenotype than if you knock down SYNGAP1, meaning that in this case, things could go too slowly, meaning that the synapses and the circuit in general would develop, let's say, instead of I mean, this is very speculative, but it would mean that, instead of taking 2 years, it might take 4 years to get to the same result, or in the mouse from 6 weeks, it could take many more weeks. So, and obviously, as always in biology, balance is really fundamental. and too much of anything is not good. So I don't think, at least in early development, it seems like a valid option to, just say, let's reduce SRGAP2A and everything will be fine. It's not like there's a good gene and a bad gene. It's really the balance between those two that's interesting. But I think on the other hand, it's really important to think that we found here probably, a sort of symmetrical regulator of synapse of SYNGAP1, sort of a twin brother, or you call it as you will, or the yin and yang brother of the synapse formation. And I think when it comes to therapy, it's usually, in many other aspects where we have made much more progress, like endocrinology, cardiology, and so on, it's actually really crucial to know about the two aspects to see on which one we can work on. So I don't know to which extent SRGAP2A will become, let's say, a pharmacological target, to be specific, but I think it's certainly very important to understand that SYNGAP1 is not just on its own here, but that SRGAP2A is next to it. And on top of it, that there is a very special type of gene that's only present in our species, SRGAP2C, that you cannot study in the mouse, and that I think justifies a lot of the experiments that we do, that I think to understand fully what goes on in the human neurons, at least in this disease, we also need to look at human neurons in very specific ways to really make sure that we fully understand the picture. To go back to your second question, so about the plasticity and the repair. So there is actually, so I showed you several times those human neurons that seem to be able to respond to visual stimulation in those mice. Now, okay, you may say, okay, this is very nice, this is like a cool gadget and that's it. Well, I would argue on the one hand, I think it's a very interesting tool to study human disease, and I hope that I convinced you of that, because we are able now to actually study human neurons deficient for SYNGAP1 in action and understand what's going on. So I think that's quite unique, and it's the early days, of course. But there's a second thought, I think, that may have come to you looking at this, is that, as I showed you, that these human neurons take many months in the mouse to develop this. It means that they develop this capacity to integrate in the visual circuit and become functional in old adult mice. So that means that they're able to work in that circuit after and long after it was established. And so I think this has very interesting implications for the prospect of basically, for instance, rejuvenating cells or transplanting new cells in a diseased brain and ask what's going on. Now, this may not be relevant for neurodevelopmental disease like SYNGAP1 but you can think, for instance, of neurodegenerative diseases, stroke, and other types of disease, where Parkinson's disease, where, as you know, a lot of researchers and clinicians are thinking of hoping to basically slow down the course of disease by adding new types of neurons in a circuit that is losing neurons. And so this, I think, is very interesting, because it shows us that juvenile neurons are able to integrate and become functional, despite the fact they are in a circuit that is already functional. And to my knowledge, this is actually the first time that this is clearly demonstrated like this. On top of that, I didn't have time to show you, but we have started now new types of experiments, completely unpublished, where we are testing now the plasticity of the transplanted neurons. So basically, the way we've been doing this is to look at what we call visual plasticity. So we change basically the visual stimulus in the mice, and we ask how the human neurons respond to this. And interestingly, what we find so far is that the human neurons display what I would call juvenile properties. So they're still very plastic, despite the fact that they are in a mouse adult cortex that does not show this plasticity anymore because they're adults. So suggesting that indeed, if young neurons, whether from transplantation or genetic manipulation or pharmacological manipulation, are found in the context of an adult circuit, it could actually lead to changes in the plasticity of the circuit. But again, this is the early days, and we'll have to see how it goes. I hope that sort of answered in part your questions.
It did. It did. Thank you. It's utterly fascinating. So we have two questions in the Q&A, and I've allowed both people to speak. Dr. Courtney, do you want to ask your question?
Yes. So I was really interested in those very large changes you showed in synaptic SYNGAP levels after the knockdown. Is that also the case for the total level of SYNGAP in the neurons, or is it about the translocation of SYNGAP1 into the synapses?
Yeah, great question. So we see these changes in synaptosome fractions. These are the data that I showed you, and we don't see that in the total levels. So we think that it's likely to be linked more to the regulation of the synaptic levels and potentially indeed translocation. You probably you know that indeed SYNGAP1 can change dramatically of subcellular localization at the synapse or outside the synapse, depending on plasticity paradigms, for instance. So I think it's very likely that this tug of war, so to speak, is really happening only at the level of the synapse and perhaps in response to activity. That's a very interesting question indeed.
And was the antibody specific to any isoform, or was it a pan-SYNGAP?
Yeah, that's another great question. This one was actually recognizing all of them, and we should definitely look into the various isoforms. You probably, so obviously you know about this. So you probably noticed on the Western blots that there are several bands. So that would suggest actually that there's more than one isoform. We've tried to look whether one would change more than another, but frankly, we have no evidence for that. I think we would need actually specific antibodies to address that question.
Yeah, it could be phosphorisoforms or something else and so on.
As well, yes. Yeah.
Okay, thank you.
Yeah, you're welcome. So
Mike, you're muted.
I was just saying, Dr. Courtney works in Finland on does some research on SYNGAP mutations. Okay. So we're very lucky to have him. JR, you had a question.
Yeah, thank you. What great research. I'm so interested in all this. I have a sort of a question, not exactly about the data you showed, but I'm interested in your visual stimulus assay and why you chose that in particular. So our SYNGAP children seem to have a strong visual system. So they have like a good visual memory. They, vision is really strong for them. Whereas like listening, sort of audio input is a lot harder. And I was wondering if you tried other stimuli besides visual, or if is that just like something pretty easy to do? Is that why you did it? Or did you try other things that didn't work as well?
I see. Yeah. So, I was not aware actually of what you were saying about the stronger visual system. That's quite interesting. So no, we chose basically the visual system, I would say, for very practical reasons, that this is a system that's relatively easy to stimulate with the mice, and where we can actually stimulate the mice with complex patterns. So while, for instance, with somatosensory or auditory, it would be much more complicated to do. So it's really mostly a practical reasons. And for the same reason, actually, we have not checked other types of stimuli. So that's why we chose the visual stimulus. There's actually another reason, which is that the human cortical neurons that we generate here from pluripotent stem cells, they tend to, so you know that there are many areas in the cortex, visual, auditory, somatosensory, and so on. And each of them has a different molecular identity. And the neurons that we generate in the dish, they tend to be more visual-like. So they tend to be basically more, potentially at least, more adapted to the visual cortex environment. So that's the other reason why we focus on visual stimulation. But obviously, it would be quite interesting to look at others.
Well, since these neurons for our SYNGAP patients seem to sort of grow up too soon, I wonder if that's, I wonder if their visual, abilities have to do with those are the neurons that develop first, right? So if those neurons develop well, they were able to develop properly before they matured, right? Whereas if
that's an interesting idea. Yes, another idea I didn't have actually. So it's in fact the prefrontal cortex, so the very front of the cortex, that is the last to develop, and the visual cortex tends to develop at earlier stages. So I guess that would sort of fit with your hypothesis. I think it goes back, but I'm sure you've been studying, I mean, you've been talking about this together, and that clinicians are studying this. I think it raises, at least to me, the question basically of the precise properties of the sensory abilities in SYNGAP1 patients. Indeed, I think it really raises the question: is there better or worse capacity in terms of responsiveness to particular types of stimuli and so on? And basically, how do the sensory system develop in general, considering what I showed, which is that some aspects may not develop at the right pace, and yet, as I showed you, at least at the particular time point where we looked at, the capacity of individual neurons to respond to, at least to discriminate different orientations and direction, was actually quite normal. I mean, actually completely normal. So I think that there's, again, more to understand, and where I think physiology and psychophysics work with clinicians and patients would be very interesting to confront with the findings in the very artificial system that we've been looking at. If that answers the question.
Yeah. Thank you so much. I have a second question that's completely unrelated. So I loved your protein blots. They were just so beautiful. And I noticed that the PSD95 was staying at the same level the whole time, right?
Yeah.
So SYNGAP was really going up and down, and it had, I mean, dramatically, and you had some upstream effectors that changed that. Have you found other? Are you studying any other genes? Like, is SYNGAP going to kind of be like a bottleneck for other maybe ion channel genes, or other other things that might be upstream? Is this going to be a bottleneck where if you actually increase SYNGAP, you could change more than one neurodevelopmental disorder?
That's another great question. So as you probably saw, I think we were very lucky in a way that we looked at very few proteins at the synapse, and SYNGAP1 turned out to change that dramatically. And in fact, to be completely honest with you, we were already, but you probably guessed that, looking at SYNGAP1 in human cortical neurons and wondering, what they could do with developmental timing. And somehow this is why we had our attention, let's say, triggered. And that's why we tested them in the SRGAP2 context. But then we got, I think, really lucky to see this dramatic result, while, as you noticed, many other proteins don't seem to change. That said, and I think that could be interesting as well, there is work that is actually also published as a preprint on bioRxiv that you may want to check, from the group of Cécile Charrier. So the group I mentioned, so she was a postdoc in Franck Polle's lab. She's now at ENS in Paris. And what Cécile did, first using mouse work, looking for interactors of SRGAP2, is on the one hand, like us, if you will, but completely independently from another perspective. She also found SYNGAP1 as a potentially interesting interactor. And another one, so that's to answer your question, what else could there be there at the synapse level that would be very important? And the other very important protein, very interesting protein that she found is encoded by, it's called basically catenin delta 2, so CTNND2, which when mutated on one allele, so haploinsufficiency of that gene is actually responsible for this syndrome called Cri-du-Chat that leads to intellectual disability and other cognitive phenotypes. It's, of course, a different disease than the one caused by SYNGAP1 haploinsufficiency, but it's also a neurodevelopmental disease and a disease of the synapse. And it's very likely that we have not tested yet, but that it could also, as a result, impair the neoteny of the human neuron. And it will be very interesting in the future to see how much, in the very simplistic model that I showed you with only 3 proteins, SRGAP2C, SRGAP2A, SYNGAP1, whether other proteins like this one, CTLND2, actually work together or against to fine regulate basically the speed at which those synapses develop and function. But I think we are really only at the beginning here. I think another very important experiment that we should try to do in the future, but that's difficult, is to really do a less biased approach, I would say, where we would try to look at all the protein interactors of SYNGAP1 and SRGAP2, but in human synapses in cortical neurons in development. So this has been done in the mouse, but never to my knowledge in the human. And given the results we got with SRGAP2C, I think it would be very interesting and important to know if there are, let's say, human-specific parts of the interactome, as we call it, so all the proteins interacting one another, and that somehow regulated the synapses. So I think that's a very, it's a big technical challenge, but it's possible given that we have an experimental model. And I think that's something that we should definitely try to do in the future.
Wow. Well, your experiments, I was amazed at how long some of them take with 18 months time and stuff. So thank you very much for working hard on this. Our kids obviously develop over many, many years. So
sure, I will definitely relay that, to actually the people who do the real work, in particular Batista, who's been really amazing to get these experiments going. They are indeed, thank you for acknowledging that they are actually quite challenging technically. Yeah.
I think, so I just shared in the chat for any of the parents who are on a link to your paper on bioRxiv. I hope I got it right. And I also shared a link to a paper that came out of Montreal last year, where I think a Spanish postdoc did some work on audio simulation, which might just be interesting since that came up. As far as the interactome and protein-protein interactions, I don't know if you're aware of Marcelo Coba, but Professor Coba is, or not, Dr. Coba is at USC, and he does a lot of proteomics and has a number of patient-derived cell lines in his lab. So he might be someone to, and I'm happy.
Yeah, I think he actually got in touch with me, and if I'm not wrong, and I think we should definitely talk. Yes,
because he's got all those spaghetti grams with all the genes, and he thinks about. He's constantly talking about genes I've never heard of, which is great. It's great. I just want to.
Of course.
No, please go ahead.
No, it was just mentioning, but I mean, so that's the difficulty of the type of research we do, right? Is that we try to go very much in depth in looking at one thing, but the price to pay for that is we cannot look at everything. And so indeed, as you saw, it takes years basically to look at one type of neuron with one deficiency. And I think the challenge we have is that, unlike, let's say, experiments in flies that takes 1 week and where you can look at many genes at the same time and get a lot of information, here we get actually information that I think is as close as it can be maybe to normal and diseased human brain, but at the same time, it's, very low throughput and very slow. So I think it will be interesting in the future to look at different mutations, to look at the various isoforms also, as somebody mentioned. But yeah, we will need much more time, I guess, to get into that. But I wish we had even more actually to more time to be able to do it faster. But it will be important in the future indeed, because we're only looking here at a very small part, let's say, of the picture.
One of the things I think about, so when we get diagnosed as families, we don't think of ourselves as in the genome. We think of ourselves as genetic epilepsies and neurodevelopmental diseases. And the big ones, like, is basically Dravet, SCN1A. And that's an ion channel. And when I'm watching your presentation and seeing how you think about, okay, this gene, actually they antagonize each other and it's about balance. I feel like the synaptopathies, SHANK3, SYNGAP1, STXBP1, are almost more complicated than the channelopathies, because the channelopathies, like, if you can just figure out the channel, and maybe I'm making that up because it's a bit self-serving, or actually the opposite of self-serving, but the balance and the network and the interaction of all these genes over the life of a human is just so complicated, and that's what you're starting to untangle, right?
Yeah. I agree with you, although I think channelopathies are quite complicated as well, and they're actually a very fascinating avenue of investigation to understand the specificity of the human brain development and function. Because, I mean, the reason why I'm saying this is that, you know, the textbook view, let's say, of channel work is that, you know, this is really like the hardcore machine of the neuron. So this is going to be the same in every neuron you look at, and this is going to be the same in every species and so on, right? And it's more the fine tuning, like at the synapse level and so on, that things are going to be different. What we found actually recently, so what other groups have found recently actually is that when you do electrophysiological recordings of human cortical slices coming from surgery resection following epilepsy, for instance, this is the work, for instance, from the Allen Brain Institute, you find actually interesting intrinsic differences in the properties of the neurons in the human versus other species, suggesting actually changes in the channels. And we recently came across another family of human-specific genes that's called LRC37. That's another preprint actually that's online, that we found is a regulator of voltage-gated sodium channels. So like the most famous, the most conserved channels of all, the most important one that makes you fire action potential, it's actually regulated in human neurons in a different way than the other species, because there's this gene LRC37 that acts on them. Again, I think it's food for thought to understand human neurodevelopmental diseases, because what we take for granted as being purely conserved may actually turn out to display differences depending on the context of the species that you're looking at. So I think in that sense, unfortunately, nothing is simple, even the channel.
No, nothing is simple. But I mean, for instance, one of the problems we have, right? 80% of our diagnosed population have protein truncating variants.
Yeah.
If you look at predictive models, like I think it's Lopez Riviera 2019, the predicted mutations you would expect to find are majority missense, minority protein truncating.
Yeah.
Right. And so why are we not getting more missense patients diagnosed? Maybe the phenotype is so severe it's lethal. Maybe it's so mild they're not presenting disease, or maybe people have no idea what to do with those mutations, and the patients are getting identified, tested, and diagnosed with variants of uncertain significance, and therefore we're not finding them because the families aren't being told, which actually happens. And so the question becomes, how do we do an assay on all of these missense mutants that we found in humans, but we don't know if they're disease causing? Are they indeed nulls and therefore creating hets, or are they dominant negative? In which case we shouldn't give them upregulating medicines, right? So this is the wall I've been banging my head against. And where ion channels, I totally agree with everything you said. Where ion channels feel simple to me is people say, well, if it was an ion channel, we just throw it in a hex cell, do some electrophysiology, we have an assay. Very nice.
I see what you mean. Yeah,
but with SYNGAP,
that's very true.
We have no way of figuring it out. So since you seem to think deeply about these things, I just want to plant the seed. If you happen to think of an assay we can do to test different missense mutations, please let us know.
Okay. No, I got what you mean. I understand better. That's totally true. In fact, and indeed, with this gene we've been studying, it's been fairly easy to do molecular assays because it's patching, and so indeed, it's way easier than look at. I think I totally see what you mean, and indeed, we'll think more about it.
Please do.
But I fully agree with you, and I mean, since the beginning that we got interested in SYNGAP1, we were very struck by the complexity of the protein, the fact that there are all these isoforms that seem to have different and sometimes completely opposite effects. I think it makes the interpretation of the genetics much more complex. And if you ask me, I think more people should study SYNGAP1 and its isoforms and so on. And I hope that putting it in the map of human evolution, if you will, is going to attract also more people to understand more what is going on. I mean, I think the fact that it's linked to SRGAP2A was a real surprise to us. Despite the fact that so many things are already known. So I think, in addition, let's say, to the type of readout you were talking about, so trying to have like higher throughput, I think that's very true. But I think another thing that we need to understand better is within the synapses, what are the molecular partners, the most critical ones of SYNGAP1 during development and so on. And there are new tools like BioID, for instance, proximity labeling, that I think would be very interesting targets for these types of proteins, where you can basically, I don't know if you know this technology, but it basically labels the proteins that are not necessarily binding directly, but that are in the neighborhood, and that can be purified on that basis. I think this is a new powerful way to understand how protein function, because at the end, how they function depends on what are the proteins that they interact with. And so this is really a new way to look at it, where I think for proteins like SRGAP2 and SYNGAP1, probably important discoveries could be made.
Amazing. Professor, Dr. Courtney has a question too. Coming in from Finland. Do you want to ask a question, Dr. Courtney?
Yeah, just related to the point, Mike, you were making about being able to evaluate different missense mutants, for example. So, Pierre, the assay you were describing with the transplantation seems to, as you said, it's a kind of cell autonomous behavior. And the cells, I don't know if they're interacting with one another, or that, there's so many mouse neurons there that there can't be that much of a direct interaction between the different human neurons you transplanted, maybe. So does that mean you could run a kind of pooled screen for multiple different mutants at the same time in the same brain? If you would label them with different fluorescent proteins, for example, you'd be able to, instead of having to do one mutant for every brain, you could actually test 10 or 20 or 100. Is that at all feasible, or is that?
I see what you mean. Yeah. So, in principle, it could be, of course. So to answer your question, so we have looked at that in the past. And so we have indeed a lot of evidence that suggest that the main interactors, synaptic interactors of each human neuron are mouse neurons. Yeah. And actually that's what makes them so functional, is that they are embedded in a fully developed circuit and so on. So that's what makes them work in a very different way that if they were, let's say, in an organoid or in a petri dish, where you would have all kinds of neurons that are quite immature interacting with one another, while here they're sort of helped, if you will, by the host. Now, we have been trying actually to do a very simplified setup of what you suggest. So basically taking normal control human neurons versus mutated haploinsufficient neurons and transplant them with a different fluorescent protein. And so that can work. The problem is that there are not so many neurons, and so you diminish basically the number of neurons that you can analyze by 2. And so if you were doing it the way you suggested, with 10 different ones, I'm afraid you may end up with only a handful of neurons on which you would need to have a readout from a screen. So I think, unfortunately, I don't think in that sense, I don't think that the system that I described is a valid or let's say realistic way to screen. I think it would be, on the other hand, an interesting system, and I think a valid system, to confirm or test in depth a particular hit, let's say, of a screen that would have occurred in vitro. And in fact, this is something we're doing for other neurodevelopmental disease, where we have collaboration with some groups basically involved in finding small molecules that can change the outcome of particular phenotypes in vitro, and where there we can use our model to look at whether these molecules have the same impact on human neurons in vivo. So as a sort of pre-translational test, if you will. But I think to find new targets, to find new hits, I think that would just not be feasible given the relatively low throughput of the experiments and also of the number of neurons we have in each mouse, unfortunately.
Okay, thanks a lot.
Thank you very much. Thank you very much. This was a fascinating presentation. I'm grateful for you.
Thank you. Thank you. Well, thank you for the invitation. I really enjoyed it very much. I hope this was clear enough for everyone. And we'll keep posted. we'll keep each other in touch to see how things move on with the field.
Please do. We're so grateful. Thank you for your time.